So today we're going to talk about light microscopy, how I observe the cells. As usual, I leave reading the introduction for you. I urge you to do that. It's important because the information there will be critical to help you to answer the questions at the end of the lab. This is just an example of the role of microscopy in a clinical pathology. So you can see different biological specimens from two patients, patient without cancer and patient with cancer. So it is a valuable tool to identify potential problems. And first we're going to talk about a specific type of a microscope, which is going to be a dissecting microscope. It's not here. So this is a dissecting microscope right here. It appears slowly, but it does appear. It's good. Okay, finally, we've got our dissecting microscope here. So you can see that there's no stage for the dissecting microscope. It is used to observe rather large specimens like a surface of your hand or some surface of an insect, a leaf, or a bug. The magnification of a dissecting microscope is usually much lower than magnification of a light compound microscope. It's often used, for instance, to perform surgeries on animals when you don't need an enormous magnification. Now, here is the compound light microscope. So why light microscope? Because it uses light, not electron beam, not anything else, light. Compound because it has multiple objective lenses. So you position the slide here and you look through the slide, not on the surface, but through the slide, because light shines from here. Okay, so I don't know why this is labeled 3. It's not oculars, obviously, you know, it's a light source, light bulb, but it's okay. And magnification of a light microscope is much higher than magnification of a dissecting microscope. So we're going to do some of the field magnification experiments. So this is the penny observed at 2 different magnifications, and you can see that as magnification increases, the field of view, the surface of the penny that you can actually see becomes smaller. So you can easily observe this here. These are human cheek cells, and that circle in the center, it reflects the cells that we kind of interested in. So at 40x magnification, so the objective here is 4, this is what you see. If you will change the objective, you see cells better, but overall field of view decreases. And if you switch even further to 40x objective, the field of view decreases even more, but you see cells in much greater details. It becomes quite obvious here. Okay, you can see the cells at great details. And how do you calculate the magnification power? Well, let's take a look here. So we've got 40x objective here at the bottom, and our eyepieces or ocular lens are 10x. So it's 40x multiplied by 10x, which will give us 400. So if we use 100 times magnification, it's 10x ocular lens and 10x objective lens. So cheek cells, if we look at them via 4x objective and 10x ocular lens. What's the total magnification? Well, we need to multiply 4 by 10 and get 40. Now, what about bacteria? Let's take a look at bacteria. We can do it. So we're going to do gram stain. You can see a lot of dots, so let's see if we can turn the objective to 10x and see something. No, not really. I mean, we can see stuff, but it's not very clear. Now, here we can see at 40x objective lenses, it's much better. You can kind of appreciate different microorganisms, but still it's not good enough. So you do 100x, but for 100x objective lenses, so 1000 times total magnification, you need to add so-called immersion oil. So we virtually adding immersion oil, and here we can see bacteria really well. We can see gram-positive bacilli, we can see gram-negative bacilli, so it looks good. Gram-negative cocci, I'm sorry. Again, we can increase the lighting, play with the lighting here. So going back to our question, if we need 1000x, you saw that with 10x ocular lenses, we had to use a 100x objective lens. So depth of the field and magnification, the higher the magnification, the lower the depth, the less you appreciate. And finally, microscopic images are inverted. So in this image, you can see the slide with the number 20 without magnification and the same slide under the microscope. We can actually do something similar right here. We're going to remove the slide. We're going to clean the microscope. After the immersion lens, it's ready to use. And we're going to do a sample slide with the letter E. So we're going to adjust letter E, and you can see that letter E in this example is flipped upside down and left to right. This is indeed what happens with the microscope slides. They are flipped upside down and left to right. So when you move slide to the left, the image moves to the right. When you move slide up, the image moves down. Now, we can't obviously observe our skin with a light microscope, don't have, the opportunity to do so. But here are some images of the skin that is observed under the compound and the dissecting microscope. So, dissecting microscope, remember, really not great magnification, allows you to observe surfaces. So this is definitely a dissecting microscope. So you can tell it by lower magnification and less details. And this is, of course, a compound microscope because the image is basically transparent. The light goes through, magnification is much higher, and it allows you to see intricate details at the cellular level. And, of course, as you increase the magnification, the field of view decreases. Now, let's take a look at the results of the observation of hair with the dissecting and light microscope. So which photograph was taken with the dissecting microscope? Of course, on the left. Lower magnification, not a lot of details. While on the right, the photograph was taken using the compound microscope. So you see it's much bigger. You can see some details there. Now, methodologically, if you go back to the method that was used to observe the skin cells, we added methylene blue to the skin cells, but not to the hair. What's the reason? If you look at the hair, it is dark. Dark and not transparent, so it can be very clearly seen under the light microscope. But in case of human skin cells, cells are very thin and practically transparent, so we needed to add an additional color to them to increase the contrast between the human cells and the background. And that's what methylene blue does. It stains the cell and increases the contrast between the cells and the background. Now here's the cheek cells. So we can look at the cheek cells. As I mentioned, we can do it here. So those are cheek cells. So we start with 4x, okay? And we can increase, for instance, lighting a little bit. We can move them up, left, right, up and down, you know, and adjust focus. Then we're going to switch to 10x, okay? And again, we can adjust the focus a little bit. And then 40x. So we can play with the diaphragm. You see how well we can see the details of the cell. And then we're going to try to do 100x. Yeah, so you can fairly clearly see the nucleus, the cell, close the diaphragm, you know, you can move things around, see the nucleus, individual cells to some extent. And here, so which ones are cheek cells, which ones are skin cells? Well, we have seen that skin cells, these are cheek cells, okay? We can tell by their appearance. Cheek cells have that very clear nucleus, while skin cells, since they're dead, there's no nucleus distinguishable in the skin cells on the left. Both skin and cheek cells appear to be flat, and they very similar in size. We're going to finish the lab with the observation of the prepared tissue slides, again virtually, and then we're going to answer some review questions. So I have, as you noticed, I have a virtual microscope, and we're going to observe a sample of blood under low, medium, and high power objective lens. So let's get to it. We're going to pick a human blood slide, and we're going to start looking at it at that's called scanning magnification, basically just to find, okay, you see faint little cells. So low power magnification, here you go. We can increase the lighting, you can see individual cells, but you can't quite clearly see them. I want you to draw this in a corresponding window in the lab right here. Now let's take a look at them at 40x. Getting darker, so we will increase the light. We can play with the fine focus a little bit. And finally, the high power magnification lens. We need to add some immersion oil. And we clearly see red blood cells here and a white blood cell. And we can move the slide around if we want to see some other stuff. And we can make it brighter. So now we're going to remove the slide and clean the microscope. And with this, we need to review the questions for the lab number 2. The proper way to dispose of human specimens. Biohazard bag. When you move microscope slide to the left, remember, the image appears to move absolutely correct. It moves to the right, and when you move a slide up, the image appears to move down. So this is you're going to do by yourself. Ocular lens, objective lens, stage. As magnification power increases, the field of view, as you remember, decreases. Proper way to carry a microscope, look at it, only with both hands. So this, total magnification of the skin cells, remember, objective lens multiplied by ocular lens, 10 multiplied by 10. We didn't create a wet mount, so you can skip that. The specimen, where do we place the specimen on the microscope? Not on the iris, not on the objective or ocular lens, we place it at the stage. Now this, here you can clearly see cheek cells, so it should be a compound microscope to see the cheek cells. And three-dimensional structure of your skin, that will be a dissecting microscope. So that concludes our microscopy lab. And I will record a little how-to video, how to upload the lab manuals.